Pharmapal.
Structural analysis of PIM1 kinase complexes with ATP-competitive inhibitors. Welcome back to our channel. Today, we're diving into some exciting research on PIM1 kinase, a key player in cancer biology. This study, conducted at several prestigious facilities across Europe, sheds light on how specific inhibitors can target PIM1, potentially paving the way for new cancer therapies. What is PIM1 kinase? PIM1, or provirus integration site for Maloney leukemia virus 1, is an oncogenic kinase, meaning it can promote cancer development. It's often overexpressed in various cancers, including leukemia and prostate cancer. This overexpression correlates with poor patient outcomes, making PIM1 a prime target for cancer treatment. Why target PIM1? PIM1 is constitutively active, which means it doesn't require external signals to function. This property makes it a unique target for drug design. Inhibiting PIM1 could disrupt its role in promoting cell survival and proliferation in cancer. The study focused on three inhibitors, CX-4945, Rho3306, and CX-6258. Let's break these down. CX-4945. An inhibitor of casein kinase 2, CK2-alpha. Known for its off-target effects on PIM1. Currently in clinical trials for leukemia. Rho3306. Primarily inhibits cyclin-dependent kinase 1, CDK1. This study explores its off-target effects on PIM1. CX-6258. A selective inhibitor for PIM1, PIM2, and PIM3. Demonstrates a promising binding mode to PIM1. The researchers used X-ray crystallography to determine how these inhibitors bind to PIM1. They found that the binding pocket of PIM1 is unique compared to other kinases, which could help in designing more selective inhibitors. Key findings. Binding mode, CX-4945 forms hydrogen bonds with PIM1, stabilizing its position in the ATP binding site. Structural characteristics, PIM1 has a bilobed structure, with a specific arrangement that distinguishes it from other kinases. Now, let's take a closer look at the figures and tables presented in the paper. We'll break down each one to understand their significance. Figure 1, Chemical structures of inhibitors. A. CX-4945. B. Rho3306. C. CX-6258. This figure illustrates the chemical structures of the inhibitors studied. Each structure is unique and plays a different role in inhibiting PIM1. CX-4945, for example, is currently in clinical trials for leukemia, while Rho3306 is known for its selectivity towards CDK1. Figure 2, Binding interactions. In this figure, we see how the inhibitors bind to the PIM1 kinase. The binding interactions are crucial for understanding how these inhibitors can effectively block the activity of PIM1. Direct hydrogen bonds. The figure shows that CX-4945 forms two direct hydrogen bonds with PIM1, which is essential for its inhibitory action. Table 1, Data collection and refinement statistics. This table summarizes the data collection and refinement statistics for the crystal structures analyzed. Here are some key points. This table provides a detailed overview of the quality of the crystallographic data, which is vital for validating the results of the study. The findings from this research indicate that PIM1 kinase has a unique ATP binding mode, which can be exploited for drug design. The structural differences between PIM1 and other kinases make it an attractive target for developing specific inhibitors. To sum it up, this study highlights the potential of PIM1 as a therapeutic target and the importance of understanding its structure for drug development. If you found this information helpful, don't forget to like, subscribe, and hit the bell icon for more updates on cancer research.
Pharmapal.